Hello students, this is Faizan Mirza. We are discussing fluid mosaic model of the membrane that was proposed by Singer and Nicholson in 1972. This is the artistic representation that you see here of the fluid mosaic model that was initially proposed by Singer and Nicholson, yes, and it was artistically represented according to the vision that was suggested by Singer and Nicholson. So to understand the cell membrane, we have to follow what the cell membrane is made up of. So the cell membrane allows transport to occur, and all the features of cell membrane and the transport that it allows comes because of the chemical properties of the cell membrane. The cell membrane is composed up of phospholipids. Phospholipid is this type of a molecule which will have a polar head and two non-polar tails. This makes this molecule amphipathic. Polar head is because of unequal distribution of charges, and since water is also having unequal distribution of charges, water is polar, so this part of the molecule can form bonds with water. The non-polar tails, they do not have any unequal distribution of charges. Unequal distribution of charges can be represented with this symbol of delta, or you can say that or sigma, in which you can say that this is slight positive and slight negative. So in a polar molecule, these charges are present, the ionic character of a covalent bond, and there are no unequal distribution charges in a non-polar molecule. The polar head is hydrophilic, while the non-polar tails are hydrophobic. The polar head is having phosphate group, PO4 3- together with glycerol. Glycerol will have 3 carbons. It's an alcohol with 3 carbons and 3 OHs. And these are the 2 fatty acid tails, the 2 hydrocarbon tails that may have double bond somewhere in the chain, or they can be all carbon-carbon single bonds. You can see here I am showing to you a phospholipid molecule which is dipping into water. So when it dips into water, the polar head, which is hydrophilic, it forms bond with water, so it can point inwards towards water, whereas the hydrophobic tails, they point outwards away from the aqueous environment. This arrangement of a phospholipid molecule is the very foundation of the cell membrane and the phospholipid bilayer. We already have discussed that water is already polar, so polar head can form bond with polar molecule of water. Cell membrane, when viewed through a very high magnification of an electron microscope, it gives a trilamellar appearance. For example, if this is a cell, this is the inside of the cell and this is the outside of the cell, and you try to magnify this part, this is how this looks like. A trilamellar appearance whose width is about 7 nanometer. Trilamellar appearance means that there are 2 structures which are darkly shaded, and then they sandwich a lighter shaded region in the center. The appearance is trilamellar, 3 layers, but it doesn't mean that the phospholipid membrane is made up of 3 layers. It's actually made up of 2 layers, bilayer. But the appearance is trilamellar. This is the outside of the cell, this is the inside of the cell. This vision, or you can say this is visualized at 250,000 magnification, which is pretty high. So Singer and Nicholson proposed that phospholipid, in fact not only Singer and Nicholson, there were several scientists, Daniel and Devson, who actually pointed out this initially, how the phospholipids can be actually arranged in 2 layers, and Goiter and Gendel. So there are multiple scientists before Singer and Nicholson who gave the idea that the membrane is actually made up of lipids, and eventually Singer and Nicholson were the one who decoded it for us in 1972. So they said that the cell membrane is made up of, or the membranes are made up of phospholipid bilayers arranged in 2 monolayers. The 2 monolayers are superimposed on one on top of each other. You can see this is one monolayer. It is superimposed on another monolayer, forming a bilayer. And the trilamellar appearance can be explained in these terms, that these polar heads which are facing outside, they form this dark region. The polar heads pointing inside towards the cytoplasm, they give this dark region, and the hydrophobic interior of the tails pointing away from the aqueous environment, they form this light gray region as seen in the electron microscope. Singer and Nicholson, they proposed the fluid mosaic model of membrane. When we're discussing the fluid mosaic model of the membrane, let's visualize the cell, that this is the inside of the cell, this is the outside of the cell. We are magnifying this part of the cell membrane. There's tissue fluid outside and there's cytoplasm inside, and we know that both of them will have water. So how the phospholipid molecules arrange themselves? So I'm just drawing these phospholipids and some various components present in the membrane, so that we can just add the labels appropriately and try to understand what the function they perform. So you can see these are two monolayers, and they are not only having phospholipid, they are having some other molecules as well. This is an artistic representation of the fluid mosaic model. The scientific findings were proposed by two scientists, as already mentioned. So what are these molecules which I have added here? So the phospholipids can be very well visualized and can very well be recognized. This molecule with one tail here, this is a cholesterol, and this one with the flipped tail is a fatty acid or a phospholipid which is forming a kink. So this is an unsaturated phospholipid. This structure here is, as already mentioned, an unsaturated phospholipid. Let's keep on labeling these. This structure is glycolipid. Why is it a glycolipid? Glycolipid means that this is having a short carbohydrate chain attached to the lipid structure. Technically, this will have actually one tail. Glycoprotein are those proteins which are present in the membrane, and they have a short carbohydrate chain attached to them. This is a short carbohydrate chain pointing towards the cytoplasm. This is a short carbohydrate chain pointing towards the tissue fluid. So this short carbohydrate chain of glycoproteins and glycolipids, they can point either way. Here I'm drawing a transmembrane protein. What do you mean by a transmembrane protein? A transmembrane protein is one which spans the whole width of the bilayer, the entire bilayer. It is crossing the whole bilayer, both the monolayers. So this is a transmembrane protein. This transmembrane protein is a carrier protein, whereas this transmembrane protein is a channel protein. What do you mean by a transmembrane protein which is a carrier protein, or a transmembrane protein which is a channel protein? So a carrier protein will not have a pore running all along its length. It will have a binding site for a substance to attach, so that it can bring about conformational change and release the substance in the other direction. Whereas a channel protein will always have a watery pathway. It will always have an aqueous pore through which water molecules can move, as well as a selective solute. For example, glucose, or for example, sodium, or for example, potassium, or chloride, or calcium, or any solute which cannot cross through the phospholipid bilayer, can be facilitated for entering from outside the cell to inside, or can probably leave from inside the cell and go outside through this transmembrane protein. It will always have a channel running all along its length, the entire transmembrane protein. The carbohydrate is attached to the lipid here. So this is a glycolipid, as already mentioned. This is a peripheral protein. So what's a peripheral protein? A peripheral protein is a protein which is present attached to one of the monolayer. It's not present in a monolayer or a bilayer. It's just present attached to one of the monolayers. And when it is present attached to a certain monolayer, it performs some function which we discuss in cell signaling and various mechanisms which we study later on when we are discussing cell physiology. These are both the examples of integral proteins. So basically, membrane proteins can be divided into peripheral proteins and integral proteins. Integral proteins are the ones which are integrated into the membrane. These could be transmembrane, or they could just be present in one monolayer. Both of them are integral proteins. But a peripheral protein is one which is extrinsic. It is present outside the phospholipid bilayer, but attached to one side of the membrane, so that it can perform a function there. The entire region of the membrane, which I am shading yellow here, is the one which is hydrophobic. So this region is hydrophobic, and the one that I've just shaded blue, this is hydrophilic. All the hydrophilic regions of the membrane are very well soluble in water. So when they are soluble in water, they provide stability to the molecules which the membrane is made up of, and they also allow the membrane to be stabilized, and the cell stays you can say stabilized in its surroundings. All parts which I've shaded yellow includes the phospholipid tails, the hydrocarbon tails of the phospholipids, of the cholesterol. The membrane proteins, the R groups of the membrane proteins here will all be non-polar because this is in the yellow shaded region. The same goes on all for all the things which are present in this yellow shaded region. Everything here will be non-polar, whereas everything which is blue shaded will be polar, be it these carbohydrate chain, be these phospholipid, the polar heads, or be these pointed ends of the proteins which are present here, or be it this peripheral protein, or this chain, or this part of the membrane protein, or this part. So the membrane proteins are composed of, again, both kind of molecules, or you can say both kind of monomers, in which you have amino acids which are having non-polar R groups. They are present here, and the amino acids with the polar R groups are present on the surface of the protein where it is exposed to the cytoplasm or to the tissue fluid. The terms of fluid and mosaic were added by Singer Nicholson. They mentioned that the membrane is fluid. Why? Because they said that all the components are in constant motion, and all the parts of the membrane are pretty much in motion, very much like the icebergs float in the sea. And the proteins, they move about like icebergs, and the water is the ocean water are these phospholipids. So the way icebergs float in water in the oceans, similarly these proteins, they float around within the phospholipid bilayer in the sea of phospholipid bilayer. Mosaic was also added. Mosaic refers to arrangement of proteins among the phospholipids when viewed from above. We have a C phase and a P phase of cell membrane when we cause freeze fracturing, and when we look at the cell membrane from the top, the spread of these proteins on the surface, it looks like as if an arrangement which is similar to mosaic pattern. This technique is applied, and when we view freeze fracturing cell membrane in scanning electron microscope. That's it from my side. Thank you so much.